Welcome to This Explainer. Today, we're diving into one of the most repeated and, honestly, least accurate claims about psychedelics and the human brain. We're going to strip away all that internet hype and look at the actual grounded science of what's really happening in your head when these substances are introduced. You know the claim. We've all seen the headlines, heard the interviews, maybe even chatted about it with friends. This idea that these substances literally grow brand new brain cells. It's basically become gospel in the wellness space. But is that actually true? Is your brain really just manufacturing brand new cells after a dose of these compounds?
Well, let's dive right in. The short answer? Mostly no. But hey, don't let that disappoint you, because what the hardcore science actually shows is way more fascinating, and honestly, a lot more useful for our mental health. Your brain isn't really acting like a factory pumping out new cells. Instead, it's doing something incredibly dynamic to the cells you already have.
To really get this, and to understand why researchers actually had to invent a brand new word, psychoplastogens, for these molecules, we have to clearly separate two concepts. We need to separate manufacturing brand new hardware from upgrading our existing wiring. These two ideas get mashed together in almost every popular article you read, but untangling them is absolutely crucial here.
All right, first up, neurogenesis. This is the literal birth of genuinely new neurons from neural stem cells. The thing is, in adult mammals, like us, this is basically confined to one tiny, specific region of the brain, the dentate gyrus of the hippocampus. It's a rare, difficult, and highly contested biological phenomenon.
Now contrast that with structural neuroplasticity. This is the real game changer. This involves zero new cells being born. Instead, your brain's existing wiring gets powerfully rebuilt. Your current neurons literally remodel themselves, sprouting new branches and these tiny little knobs called spines, where fresh synaptic connections form. So when people say psychedelics rewire the brain, it's this second mechanism, lipid plasticity, that the solid science actually backs up.
Section 1. The proof of plasticity. Rapid rewiring. Almost all the robust evidence points strictly to this rewiring effect, not new cell growth. Let's look at the proof. There was this landmark 2021 study out of Alex Kwan's group at Yale, where they actually watched this happen in a living brain. Using advanced two-photon microscopes, they found that a single dose of psilocybin bumped up dendritic spine density in a mouse's cortex by roughly 10% in just 24 hours. Just imagine that for a second. Your brain's communication network physically expanding its connection points by a tenth, literally overnight.
And what's wild is how durable this is. That rapid 10% growth happens in a single day, sure, but crucially, when they check back a full month later, those newly built neural connections were imaged and found to still be physically there. This rapid, very real, and sustained rewiring after just one dose, that is the absolute defining signature of a psychoplastogen.
So how exactly does this happen deep inside the cell? It's a pretty fascinating twist, actually. Because psychedelics like LSD and psilocybin are fat-soluble, they just slip right through the neuron's outer membrane. Once they're inside, they hit a hidden pool of 5-HT2A receptors that our brain's normal, water-loving serotonin literally can't reach. They also seem to directly bind to another receptor called TrkKB. Both of these entry points act like molecular trigger switches. They kick off a specific biological cascade known as BDNF to TrkKB to mTOR, which fires up the protein synthesis that physically builds those new dendritic spines.
Section 2. The Skeptic's Corner. A scientific reality check. Now you might be wondering, wait, if growing new brain cells is mostly a myth, where on earth did all those headlines come from? Impartially speaking, the evidence for brand new cells being born is incredibly weak. It mostly stems from preclinical studies. We're talking mice and cells in petri dishes. In fact, one study showed that while low doses of psilocybin kind of trended toward an increase in newborn neurons, high doses actually decreased them. And yeah, there is a study showing the ayahuasca molecule DMT stimulates neural stem cells, but it does so through a completely different pathway called the sigma-1 receptor, not the classic psychedelic 5-HT2A pathway. On top of all that, this entire idea rests on a massive, unresolved debate over whether the adult human hippocampus even grows meaningful amounts of new cells anyway. I mean, in 2018, two major papers reached complete opposite conclusions on this exact question. And things get even tougher for the human molecular proof when we look at a recent, massive meta-analysis of 29 different studies. Researchers specifically looked at blood levels of BDNF, which, like we just talked about, is the crucial growth factor at the very heart of the brain's plasticity cascade.
The results? There was absolutely no overall effect of psychoplastogens on blood BDNF levels. Just zero.
Now look, this doesn't mean the rewiring isn't happening, but it hits us with a really vital scientific reality check. While animal brain imaging is incredibly sharp and clear, a human blood test is a totally inadequate window into the rapid, microscopic changes happening deep inside a living human brain. The molecular confirmation in humans is still just an educated guess, not a settled fact.
Section 3. The final takeaway. The window of change.
So, if these drugs are mostly rewiring us rather than growing new cells, why is that such a game changer for things like depression, PTSD, and addiction? Well, a striking 2023 study by Dolan's lab paints a brilliant picture here. It turns out psychedelics seem to reopen a juvenile critical period of heightened learning. Basically, they briefly hand your adult brain back a superpower and mostly lost after childhood. And what's absolutely wild is that the duration of this open window scales directly with how long the drug's subjective effects last in humans. For ketamine, where the trip lasts a couple of hours, that critical plasticity window stays open for about 48 hours. But for a drug like ibogaine, where the subjective effects can literally linger for weeks, that window of profound brain malleability stays open for weeks too.
The absolutely crucial takeaway here is that this rewiring is not some magic cure-all. You can't just take a pill and get a permanently fixed brain. It doesn't work like that. Instead, you've got to think of it as a temporary state of heightened malleability. It's a precious, fleeting window where your brain is unusually capable of overriding deeply entrenched patterns, stubborn habits, and traumas. And this is exactly why researchers constantly hammer home the importance of therapy and integration alongside the medicine. The drug, it just opens the window. But because your actual experiences in your integration are what determine the durable physical changes built into your neural wiring, we have to end this explainer by asking you one thing. Once your window of malleability is blasted open, what will you choose to build? Thanks for joining me today, and I hope this brought some real clarity to how your brain actually works.